Hi everyone, this is Ms. Romani, and in this lesson we will be learning about photosynthesis. And photosynthesis is a process that converts light energy into the chemical potential energy that is found in the bonds of sugars. And not just glucose, but all the sugars that plants produce, like fructose or starches, and even cellulose. In the cellular respiration lessons, we learned how living things can store the energy in sugar bonds into the energy found in the bonds of molecules called ATP. And if you recall the overall chemical reaction of cellular respiration, then you may actually notice that the overall chemical reaction of photosynthesis, seen here, is basically the opposite of that reaction, with carbon dioxide and water as the reactants instead of the products. And just like with cellular respiration, the process of photosynthesis is not completed as a single reaction, but as a metabolic process that has many steps and individual reactions, which will be covered during this lesson. In most plants, photosynthesis usually happens in a structure called the leaves, and leaves are actually a perfect shape for photosynthesis to occur. They are filled with these types of cells called mesophyll cells, but also their flat and wide shape allows them to maximize sunlight that can reach the cells, and it allows for carbon dioxide gas to flow very easily right through the leaf surface into the cells and for them to excrete oxygen gas out of the cells. They have these little pores, usually on the underside of a leaf. These pores are called stomata, and they open and close and allow carbon dioxide gas and oxygen gas to easily be exchanged. The mesophyll cells themselves are filled with these types of organelles called chloroplast, and it is in this chloroplast that photosynthesis takes place. So let's take a look at the chloroplast in a little bit more detail. Within each chloroplast, we find these disc-like structures called thylakoids. Now, thylakoids I like to describe almost like pancakes, but unlike pancakes, they are hollow on the inside. So there's actually a little bit of space between the top of, let's say, the pancake and the bottom of the pancake. It's kind of hollow in the inside. Now, the reason why we like to describe them as pancakes is because they're arranged usually in stacks, like pancakes are arranged in stacks, and the stacks are called granum, plural grana, singular granum. The membrane of each thylakoid contains the green color pigment called chlorophyll, which is what gives both chloroplast and plants their green color, and it is this chlorophyll that can absorb light. The stacks of grana are basically found within this fluid-filled area of the chloroplast called the stroma. And the stroma is just basically the space inside the chloroplast that is not the thylakoids. But within the thylakoids themselves, there's another fluid-filled space. This one is obviously much, much thinner, and that's called the lumen. And so the lumen is the space that is found inside those hollow pancakes that we call thylakoids. So last week we learned that cellular respiration happens in 4 stages. The first of which, glycolysis, happens in the cytoplasm and not inside a mitochondria. Well, photosynthesis also happens in stages, but only 2 stages this time, both of which happen in the chloroplast. The first stage is called the light reactions or the light-dependent reactions, and this stage actually involves an electron transport chain. While cellular respiration ends with an electron transport chain, photosynthesis actually starts with one. And during these light-dependent reactions, the point of them is basically twofold: to produce molecules of ATP and to produce molecules of NADPH. And no, that's not a typo. I did not mean to say NADH. NADPH is just a different type of electron carrier, just like NADH and FADH2. And the light energy is also used to split water and to release oxygen as a byproduct. And the electron transport chain where the light reactions happen can actually be found in the thylakoid membranes. The second stage does not need light and is called the light-independent reactions. Sometimes they're called the dark reactions, but that's a bit inaccurate because that might imply that they can only happen in the dark. But these reactions can happen in the light or in the dark. They just don't need light. The light-dependent reactions actually need light. The light-independent reactions don't, but they can happen in the dark or they can happen in the light. It doesn't matter. The light-independent reactions are basically a series of chemical reactions collectively known as the Calvin cycle, and their whole purpose is just to form sugar, to form carbohydrates using carbon dioxide gas from the air. And they take place in the stroma of the chloroplast, not in the membranes, in the stroma, in that empty space in between the thylakoid stacks. So let's start by examining the events of the light-dependent reactions. But before we dive in, I want to just quickly review a little bit about the nature of light. And light, as you may already know, is a type of energy. It is a form of electromagnetic radiation that travels in packets called photons. And those photon packets have specific wavelengths depending on the amount of energy they possess. So the light that we see is called visible light, and visible light has more energy than, say, radio waves or microwaves, but they have less energy than, say, UV rays or X-rays. And chloroplasts have molecules called pigments that help them absorb that light. And the one that you may have heard about the most and which makes plants look green is called chlorophyll, but there are actually other pigments. They're called accessory pigments. The carotenoids, for example, are a type of accessory pigment that give plants the yellow and orange colors that you often see when the leaves change color in the fall. And as a matter of fact, those colors are always present in the leaf. It's not like that the leaves turns colors by having their pigments change or new pigments appearing, mostly. It's just that the carotenoids are always there right underneath the green, but there's always so much more chlorophyll present that the green masks the orange and the yellow. So it's only when the plant starts to break apart the chlorophyll, which happens when the days get shorter and there's a lot less solar energy, and that the plants start to basically break apart the chlorophyll in order to conserve energy in the cold, that the colors underneath, the greens, the oranges and the yellows are sort of revealed. And so different colors have different wavelengths and different amounts of energy. So when we look at an object, the color that we see is determined not by the wavelength of light that it's absorbed, but by the wavelengths of light that are reflected back from that object. So that means that when we look at something green, like say a plant, the color of light that is absorbed the least by the plant is the color green. That's the color that is reflected back to our eyes, and that's the color that the plant appears. So this graph right here shows us the different wavelengths of light that are absorbed by different pigments found in plant cells. So notice that the color green is not absorbed by any of the pigments. The green is there between the wavelengths of 500 and 600. And notice that there's no pigment that is absorbing those wavelengths of light. And that is why plants appear green to our eyes. Those wavelengths of light are reflected back to our eyes. And that's the color that plants appear. So for example, if you were to place a plant in a room with only a green light bulb as a source of light, the plant would be unable to obtain energy and would die. In the chloroplast, we will find the pigments organized in a structure called an antenna complex. This antenna complex is actually made up of a type of chlorophyll called chlorophyll A and a bunch of another version of chlorophyll called chlorophyll B and the carotenoids. So notice that in the graph picture that the different pigments absorb different wavelengths of light. Chlorophyll A, chlorophyll B, and the carotenoids all absorb different wavelengths of light. So by combining different pigments, the antenna complex can then maximize the amount of energy that is absorbed from sunlight. And like an antenna, it gathers all the energy and transfers it to one central point. The central point is called the reaction center, and the molecule found in this reaction center is a molecule called chlorophyll, very specifically chlorophyll A. So what happens when the concentrated light energy is then absorbed by a molecule of chlorophyll A? Well, the energy from the light excites a pair of electrons from their ground level to an excited energy level. This high energy electrons can have several fates. For most compounds, the electrons would simply return back to their ground state and the absorbed light energy would be converted into heat. However, if a suitable electron acceptor is nearby, like in the case of photosynthesis, the excited electrons can actually move from the initial chlorophyll molecule to the acceptor. This is actually the start of the electron transport chain of photosynthesis. So the whole antenna complex and the pigments, including chlorophyll A, are found within these complexes of molecules called photosystems. So let's take a look at these photosystems and the rest of the electron transport chain of light-dependent reactions in a little bit more detail. Okay, so I want you to bear with me for a minute as we go through this slide. It's going to take a little while. So what we're going to do is we're going to go through the entire process of the light-dependent reactions, starting with light hitting the antenna complex and then moving all the way up until the production of both the ATP and the NADPH that are the products of this first stage of photosynthesis. So I'm going to take you step by step through this. If you need to rewind the video and watch parts of it again, please do so as often as necessary. You should also have a blank copy of this sheet in your handout. Don't start filling it in right away. Maybe watch through it the first time, and then maybe if you want to, you can fill in what is happening as you move through the video, maybe a second time. And then you can maybe use a blank copy of that just to sort of review and study for your test to make sure that you actually understand what's happening during the light reactions. This is the part of photosynthesis that tends to be the one that has a little bit more details and tends to be a little bit more complicated. So I'm going to go through it step by step though. So let's start at the beginning. Let's first of all orient ourselves to where we are. See the picture of the chloroplast at the very top, and there's a kind of a little circle there. So that's where we're located. So that little circle is what you see in front of you in the main part of the slide, that piece of the membrane. So we're on just a tiny little corner of a thylakoid. And so this is the thylakoid membrane, and right outside of that, it's the stroma of the chloroplast, which is outside of the thylakoids. And the space right here would be the lumen, which would be the inside little sliver of space that is inside each thylakoid pancake, if you will. Now, I've also labeled on here the two photosystems. The first photosystem is called photosystem 2, and the second photosystem is called photosystem 1. I know, it makes no sense. But that's because the photosystems were actually named after in the order basically that they were discovered, not in the order in which they appear and they function within the electron transport chain. So something we have to deal with. The first photosystem is called photosystem 2. The second one is called photosystem 1. They also have been given this weird tiny little sort of code. Photosystem 2 is called P680 and photosystem 1 P700. Now, what does that mean? Each photosystem is slightly different than the other. Photosystem 2 has a chlorophyll A molecule in the reaction center. Remember from the antenna complex, the reaction center is where the light gets transferred to. The chlorophyll A in the reaction center of photosystem 2 can absorb wavelengths of light best at wavelengths of 680 nanometers. And so that's why it's called P680. Now, as you may guess, the chlorophyll A that is found in photosystem 1 can best absorb wavelengths of light at 700 nanometers. So hence the name. P680 and P700 is based on the wavelength of light that is best absorbed by each photosystem, and photosystem 2 and photosystem 1 are named based on the order in which they were discovered. Okay, so let's begin. The first thing that's going to happen is that light is going to hit the antenna complex of photosystem 2. Remember, the antenna complex is just a series of accessory pigments that are going to guide that light or concentrate that light and move it in a concentrated form to the reaction center where a molecule of chlorophyll A is found. And so that chlorophyll A is going to take that light energy, and that light energy is going to be used to excite electrons in chlorophyll A to a higher energy level. Now, normally, when light hits pigments and the electrons in the pigments get excited, they very quickly return back to their ground state, to the original state, and the energy is basically released as heat. But this is an electron transport chain. And so the electrons, instead of returning back to the molecule of chlorophyll A, will be quickly grabbed by a very electronegative molecule within the electron transport chain, and the electrons are essentially going to be moved through an electron transport chain right away through a series of molecules. Now, notice what happened when the electron transport chain happened. Just like in cellular respiration, the energy loss as the electrons moved down the electron transport chain was used to pump some hydrogen ions or protons from one area of the chloroplast that had maybe the hydrogen ions in less concentration to a different area where the hydrogen ions would be found in a higher concentration. Now we'll get back to the hydrogen ions or protons in a minute. Let's focus right now on just what's happening to the electrons, okay? So the electrons from chlorophyll A in photosystem 2 move through the electron transport chain. The energy was used to pump some hydrogen ions across from the stroma into the lumen, but the electrons actually will end up right here in photosystem 1. They will end up at the chlorophyll A of photosystem 1. Now, in the meantime, the chlorophyll A from photosystem 2 is without electrons. The electrons have left it. So those electrons need to be replaced if we want the process to kind of continue and be an ongoing process. And to do that, we're going to be splitting some water. So this molecule right there that has a Z on it, it's a water splitting complex. And what it basically does is it takes water and removes its electrons, and in the process splitting it, releasing oxygen gas and hydrogen ions or protons. And those electrons that are removed from the hydrogens that were in water are used to replace the electrons that were lost by chlorophyll A in photosystem 2 into the electron transport chain. So the next time when light hits the antenna complex on photosystem 2, there's fresh electrons that can then move through the electron transport chain once again. Okay? So let's go back to the electrons that ended up at chlorophyll A of photosystem 1 because they're not done yet. So those electrons will also be absorbing some solar energy. The antenna complex is going to transfer that energy right into the chlorophyll A that picked up those electrons, and the electrons will be moved to a higher energy state and move through another electron transport chain, and the final destination in this case is NADP+, and the formation of NADPH. So those electrons are going to be carried by an electron carrier called NADPH. So let's get back to those protons then. The movement of hydrogen ions into the lumen combined with the splitting of water will now have created a high concentration of protons in this very thin area inside the thylakoid called the lumen. Because this area is so thin and so narrow, then a high concentration of hydrogen ions is very easy to achieve, especially because you're splitting water constantly, so then more hydrogen ions are being accumulated in that area, and electron transport chains are happening, the first of which is basically used to pump hydrogen ions into that small area as well. So what we're doing is we're creating an electrochemical gradient that, just like in cellular respiration and the electron transport chain there, that electrochemical gradient is going to be used to make some ATP. So we have a molecule that, once again, we've seen before, ATP synthase, which can be used to pump hydrogen ions across from, in this case, the lumen into the stroma. And the energy that is released by those hydrogen ions as they move down their concentration gradient from an area of very high concentration to an area of much lower concentration is used to phosphorylate ADP into ATP. And that's it. We made the two products that we needed to make. The whole point of the light reactions of photosynthesis was to make molecules of ATP, which we've done through the creation of an electrochemical gradient by the high concentration of protons in the lumen of the thylakoid. And we've reduced NADP plus into NADPH by adding high energy electrons in there. Those electrons were excited and given more energy by sunlight, and the electrons ended up in molecules of NADPH. And now these two molecules NADPH and ATP are going to be moving on to the light-independent reactions and the Calvin cycle, which is what we're going to be discussing next. So let's take a look at the Calvin cycle then. As we move through the Calvin cycle, I don't want you to worry too much about the structure of the molecules that are part of the Calvin cycle, or even the number of molecules that are involved in each step. Basically, we're going to divide it up into 3 phases, and I just want you to just keep track of what the main purpose is of each of the phases. So just sort of for each phase of the Calvin cycle, think about what's the purpose, what is the main point of each phase, and just worry about that. Don't worry so much about what's happening to the independent molecules that are moving through and how many of them there are. Those are just details that are not necessarily something that you're going to be tested on, or that is a main idea of this lesson. Okay, so let's begin. So phase 1 of the carbon cycle is what we call carbon fixation. And this process is probably one of the most important processes of photosynthesis, because this is all about taking carbon dioxide gas from the air and putting it into sugar. And the enzyme responsible for this very crucial first step of the Calvin cycle is called rubisco. Rubisco is found in all photosynthetic cells and is probably the most abundant and most important protein on Earth. Rubisco is able to basically capture carbon dioxide from gas form and put it into a sugar molecule by catalyzing that first reaction of the Calvin cycle. And in this reaction, what happens is rubisco combines 3 carbon dioxide molecules with 3 molecules called rubulose bisphosphate, or RUBP for short. And the product of this reaction is 6 molecules of a 3 carbon sugar called 3-phosphoglycerate. So you end up with 6 molecules of that. That's phase 1. It's just that first reaction of getting carbon dioxide into a sugar molecule. Okay. Phase 2 is about energy, and it's called the reduction phase. And so in this next phase of the Calvin cycle, both ATP and NADPH from the light reactions will be used to both phosphorylate the sugars and to reduce the sugars in order to increase the energy in the sugar. So the 3-phosphoglycerate is going to be first be phosphorylated by molecules of ATP from the light reactions, and they're going to be turned into molecules called 1,3-bisphosphoglycerate, because now they'll have an extra phosphate group each. And then those molecules are going to be reduced, so they're going to be gaining the electrons that came with NADPH from the light reactions, and they're going to produce molecules called glyceraldehyde 3-phosphate, or G3P. So at the end of this process of the reduction phase, we'll end up with 6 G3Ps. Each G3P has 3 carbons and 1 phosphate group. Now, this is a step where the sugar is actually made, because for every round of the Calvin cycle where 6 G3Ps are made, one of these G3Ps is going to exit the Calvin cycle, and then it's going to be used to make glucose or other sugars. So what will happen is the Calvin cycle will have to go through 2 rounds. Remember that at the very beginning of the Calvin cycle, we started with 3 carbon dioxide gases. But we know that we need 6 carbon dioxide gas to make 1 molecule of glucose. And that's because after the first round of the Calvin cycle, 1 G3P molecule exits that has 3 carbons. So we need a second round of the Calvin cycle to have a second G3P exit. And so another 3 carbon dioxide gases to have another G3P exit. And then what will happen is 2 of those G3Ps will combine and they can form glucose, or they could form fructose, and then those glucose and fructose can form sucrose, starches, cellulose. They can even be turned into proteins or fats within the cell, so any organic compounds that the cell needs. Now the last stage and last phase of the Calvin cycle is just about regenerating RUBP, just going back down to the beginning. And there are a series of reactions within that. We're not going to get into those, but essentially it's just a series of reactions that are just going to form RUBP once more from the remaining G3Ps. And then the process can begin again. Now, I want you to notice that there are 3 ATP that are needed for this phase 3 of RUBP regeneration. And that's what we're going to be discussing in a moment. How do we get that extra ATP? So, under certain conditions, the photoexcited electrons take an alternative path called cyclic electron flow, which uses only photosystem 1 or the P700 photosystem. Photosystem 2 or P680 is not involved at all. This process produces only ATP. NADPH is not produced, and actually water is not used at all, so there is no oxygen production. Now, during non-cyclic electron flow, which is the name of the electron transport chain we learned about earlier in this lesson, the final electron acceptor for the photoexcited electrons is NADP+, which gets reduced to NADPH, and then leaves the electron transport chain so it can go on to the Calvin cycle. So for that particular process, we needed a constant supply of water to act as electron donors because the electrons are leaving the electron transport chain. But cyclic electron flow doesn't need water because the electrons never leave the electron transport chain. They are excited by photons of light and move to a higher energy state and then are transported via a series of redox reactions through the electrons transport chain right back to where they started, which is why it is called cyclic electron flow. The energy the electrons lose as they move to a lower energy state is used to create a proton gradient. And that proton gradient is then used to phosphorylate or photophosphorylate ADP into ATP. Now, one of the reasons for cyclic electron flow is to generate extra ATP in order to balance that ATP to NADPH energy budget that is needed during the Calvin cycle. Because often the amount of ATP needed to drive the Calvin cycle exceeds what is produced in non-cyclic photophosphorylation. For example, that last phase of the Calvin cycle that I asked you to pay attention to required that extra ATP for RUBP regeneration. And that's it. That's photosynthesis. Energy from the sun was harnessed in the light reactions, and that was used to phosphorylate ADP into ATP and to reduce NADP plus into NADPH. And then during this process, water was split and oxygen gas was produced and released as waste. And then finally, the energy from ATP and NADPH from the light reactions was used during the Calvin cycle to fix carbon dioxide gas into molecules of G3P, which were then combined into sugars and other organic molecules. One thing that you may have noticed by now is that photosynthesis and cellular respiration are opposite and complementary reactions. The products of one reaction are the reactants of the other and vice versa. Also, an interesting thing to note as well is that unlike animals that only undergo cellular respiration, plants actually have both chloroplast and mitochondria, so they undergo both types of reactions. So they undergo photosynthesis to store solar energy into sugar, and then cellular respiration to transfer the energy in that sugar that they created themselves into the ATP molecules that the cells can then use. And that's it for this lesson. Please remember that if there are any concepts from this unit that you may not yet understand, you are encouraged to read the lesson notes or watch the supplemental videos, complete the homework questions, or just complete any of the extra activities that I've provided. All of those are there to help you understand the concepts. And of course, don't forget that you can always reach out to me for extra help. Talk to you soon.